Nice to see you again. We'll do our best to give a good overview of the paper, which is a very interesting paper. And Costa is gonna give.
Okay, I'm gonna share the whole screen, right?
Yeah, I think that would be best.
Okay, just let me know if you can see everything. And if you can see, sorry. Okay, so can you see me changing the slides?
Yes, perfect.
Okay.
The floor is yours.
Okay, so thank you for having us. So it's Filippos and Konstantinos. So we're both PhD students in the same lab, but working on completely different things. So more or less, we have things here that one works more or less with these kind of techniques, and I work with other kinds of techniques. So we kind of mix this paper. So the paper that we're gonna present it was published in December 2024, so it's a recent paper. The title is Multiscale Drug Screening for Cardiac Fibrosis Identifies MT2 as a Therapeutic Target. And the team that published it is from the US and the lab of Joseph Wu. A little bit of introduction. So cardiac fibrosis is characterized by excessive extracellular matrix deposition, stiffness of the organ, and eventual dysfunction of the heart. What happens is that in response to stress or some stimuli, cardiac fibroblasts, they become activated. One of the markers is alpha-SMA, as you know, and they subsequently produce and deposit extracellular matrix on the tissue. It's a clinically unmet disease. There is need for treatments. So there are only two FDA approved drugs against idiopathic pulmonary fibrosis, and it's the nintedanib and the pirfenidone, both of which failed to improve cardiac function in human patients.
So I know you can see the titles. Okay, so this paper actually these guys present their work in a very nice using a very nice methodology, which can be grouped into 8 core steps, let's say, which they start from some in vitro screenings, they go in vivo, and finally they find the mechanism of action of their best compound against cardiac fibrosis. So we'll try to go through each step into some detail in order to make it more comprehensible for each one. So to start off, what they did, these guys, that they used as a screening tool cell line they generated. So they have used human-induced pluripotent stem cells to generate both cardiac fibroblasts and the cardiac cells. So this gene-edited cell line, what they did for these lines, they tagged the myofibroblast marker ACTA2, so alpha-SMA, with the fluorophore clover-2 upstream of the ACTA2 stop codon using the CRISPR-Cas9 technology. And what they've shown is that upon TGF-beta1 stimulation, alpha-SMA signal, which translates to the clover-2 signal actually, it's increased, while upon pirfenidone treatment plus TGF, this signal is decreased, which is what we also see in the literature. So after establishing this gene-edited cell line, they've used a dual staining method in order to screen a 5,000 compound library. What they did is they stimulated cardiac fibroblasts with this gene editing, and they have been observing their nuclear number with host staining and the alpha-SMA expression levels through the clover-2 signal. So what they did is that through the color intensities, they could quantify the effective concentration and the toxic concentration of each compound. And based on these two values, the compounds were categorized in three groups. The first group, it was the weak or / non-anti-fibrotic compounds, which are represented by examples 1 and 2. So slight decrease in alpha mass signal, together with some toxicity, or very slight decrease in alpha mass signal with no toxicity. Second category, toxic compounds. So you might see at the example 3 decrease in the Clever-2 signals alpha mass signal, but it's accompanied with decrease in cell number. And the third category, potent antifibrotic compounds, which these compounds, this example here, example 4, there's no change in the cell number, but there is a good alteration of the Clever-2 signal, so alpha mass expression. So through this screening and some parameters they set based on the acceptable EC50 and TC50 values, plus using a machine learning tool for checking optimal ADMET properties of the compounds, they filter out 20 best compounds through which they moved on. And what they did in order to choose the best compound out of these 20 group of compounds, what they did is that they used iPSC cardiomyocytes and epithelial cells for cytotoxicity. And what they've ended up with is artesunate as their top candidate with a good EC50, 2.1 millimolar, exhibiting no toxicity to the cardiomyocytes or the epithelial cells. So artesunate, or ART as we'll be calling it, is one of the most efficacious therapies for malaria by binding heme in red blood cells to generate reactive oxygen species. ART discovery was granted with a Nobel Prize back then. So in order to reassure that indeed ART effect is robust, they've examined the alpha mass expression upon different fibrotic stimuli, as you can see here, and indeed the effect was pretty robust. In all cases, ART could reduce the alpha mass expression in cardiac fibroblasts. So they moved on checking different aspects of anti-fibrotic activity on primary healthy cardiac fibroblasts or diseased cardiac fibroblasts, including in vitro pro-fibrotic marker expression, such as collagen 1 and ACTA2, which, as you can see here, are reduced, proliferation of cardiac fibroblasts upon TGF, which is reduced both in healthy and diseased fibroblasts, cardiac fibroblasts. The migration of the cells again is reduced, the contraction of these fibroblasts again reduced upon ART treatment, and apart from the 2D screening system, they generated the 3D engineered heart tissue model, which was composed both of cardiomyocytes and cardiac fibroblasts, in order to recapitulate the characteristics of cardiac fibrosis in vivo. So because this model mimic the heart, they checked some core aspects of heart function, which are all improved, as you can see at J and K figures, upon ART treatment. And apart from these aspects of heart function, they have seen that ART treatment also reduces some of the key fibrotic markers during fibrosis, as ACTA2 and collagen 1 in this 3D model.
So the next thing in this paper is the in vivo part. So for this, they used model of heart failure that is the transverse aortic constriction model. So in this model, they performed the surgery, and one day after, they started daily treatments, IP injections of ART for 4 weeks. So at the end of this protocol, they assessed the histology, the ECM deposition, the fibrosis in histology, and also in collagen deposition. And what they see is that upon our treatment, there is decrease in the collagen deposition and also in the histology. Also in the echocardiography that they did, they see that there is decrease in the left ventricular ejection fraction in this model, as it is known, and that there is amelioration in the treated mice. Also, the same thing with the diastolic function, as is measured by the E-to-A ratio, which is also a marker for the stiffness of the myocardium. They could see amelioration here. So next, except that this is the the preventive model. They wanted to see a therapeutic model. So after the surgery, they let the mice for 4 weeks before starting the daily treatment with ART, for 4 weeks again. So again, the same results, both in histology and in collagen deposition, they could see amelioration. And in the injection fraction of the left ventricle, again, decreased. They had decreased ejection fraction in the vehicle treated mice, and in the treated mice without, they have amelioration. The same thing with the diastolic function. So they also move forward with another model. The other model is a model that recapitulates the clinical characteristics of the myocardial infarction in humans, and it's the ischemia perfusion model. So in this model, 3 days after the surgery, they started treatment for 2 weeks. Again, in histology, in Masson stainings, and in collagen deposition, they saw amelioration of the fibrosis. And in the echo, again, the same results, with the amelioration of the disease, both in the left ventricular ejection fractions and the E/A ratio. So the next step here was to see the cell populations. So what they did is, in the preventive model of the TAC, they did single nuclei RNA sequencing in the hearts. They were able to recapitulate most of the different populations, cell populations of the heart. And in the fibroblast here, you can see increased expression of genes of collagens and genes related to fibrosis and extracellular matrix. And in the treated ones, they could see decreased expression compared to the model. Also, when they saw they compare the GO terms that are related to fibrosis, they could see that it's decreased in the treated. So these GO terms are decreased in the treated mice. So when they went to the fibroblast populations, now they were able to find 6 different populations. And interestingly, this population here, the population number 1, was increased, as you can see, in the diseased mice, and this proportion was decreased in the treated mice. So this population is a population of fibroblasts that highly express genes related to fibrosis and a lot of collagens. When they ran a pseudotime analysis, they could find that late during pseudotime is the population number 1, which is in TAC mice. And this, as the pseudotime goes and the trajectory goes, it goes back again in the treated mice. So they proposed that this population, when we have the heart failure, these fibroblasts, they increase the expression of fibrotic genes, and then with ART, this expression goes down. As I told you, this population express a lot of genes related to fibrosis. As you can see here, one example that they have is periostin. As you can see here, we don't have nothing in the healthy mice. In the diseased, we have expression here that lies the population number 1, and decreased expression in the treated mice. An interesting part was that when they saw the immune cells of this model, they didn't find any changes in the macrophages, in the proportion of the macrophages during in the 3 different models, the 3 different mice. And what they propose is that ART doesn't have a role in the inflammation in this model. So the next step was to take cell lines, healthy human cardiac fibroblast primary cells, not cell lines, I'm sorry, and they treated them with TGF and different concentration of ART, and they could see decreased both in mRNA levels and in protein of fibrotic markers. The same thing with the stimulation with interleukin 11. Next step was to take disease myofibroblasts from patients. They treated them with ART, and again, they saw a decrease in the fibrotic markers. The last step here was they took cells from 3 different healthy donors of fibroblasts from the heart, and they treated them with TGF-beta 1 or they treated them with TGF-beta and ART. What they saw is that they increased genes related to fibrosis on TGF-beta 1, and in the treated ones, they saw decreased expression compared to the treated ones. And also in the TGF-beta treated ones, they have again increased fibrosis and extracellular matrix terms.
Okay. So after the examination of the anti-fibrotic potential of ART both in vitro and in vivo, they decided to go more into depth concerning the mechanism of action of this compound. So what was known is that ART can inhibit the toll-like receptor 4, TLR4 signaling in macrophages. So together with TLR4, MD2 is a co-receptor which upon LPS, it binds the TLR4 and it forms a co-receptor complex which signals that downstream. And apart from LPS, this complex can bind also ECM components which are upregulated upon TGF-beta, such as fibrinogen noir or tenascin C. So in order to examine if this receptor complex plays a role in cardiac fibroblasts and in cardiac fibrosis, they silence either MT2 or TLR4 in both healthy and diseased fibroblasts. And what they've seen is that indeed there's reduction of some key fibrotic markers, while it also affects the proliferation of the cells, deletion of one of the two of either one or the other receptors. So they did a surface plasmon resonance to investigate MT2 ART binding potential, and as you can see, it came up with a relatively okay binding capacity. In order to examine further this phenomenon, they did some in silico molecular dynamics simulation, which revealed that ART can antagonize the MT2 TLR4 interactions. So what they did is that they chosen tryptophan 23 amino acid in the MD2 structure, which is considered to be a key amino acid for the conformational changes of this protein. They fluorescently labeled this amino acid, and they have shown that upon ART binding, there's a reduction in the fluorescence signal, which means that it can cause conformational changes in the protein structure. Further in silico studies revealed ART potential to antagonize LPS binding on MT2, while comparison with L6H21, which is a known MT2 inhibitor, showed ART's improved potency to disrupt LPS MT2 binding, which is depicted by this graph here and a class score. Moving on through ELISA, they have shown that it's a specific ELISA that they've designed where they've biotinylated LPS and have shown that through time, as you can see here, and ART has a potential to replace the LPS, the binding site of MD2, even better than L6H21. Further, they performed proximity ligation assay fluorescence in 2 screening systems. So they used HEC cells overexpressing MD2 and TLR4, and they have shown that upon ART LPS treatment and ART treatment, there's reduced interaction of this complex, while they have also confirmed this phenomenon in cardiac fibroblasts that upon TGF-beta 1, there's reduced formation of MD2 TLR4 complex. Then they used attack sequencing in cardiac fibroblasts to reveal that ART can reduce TGF-beta 1 induced chromatin stability of fibrotic genes, which are shown here, collagen 1, fibronetin, etc., which are key fibrotic molecules. And through this sequencing, they have shown that AP1 family was a top-down regulated motif by ART, where both ERK and CFO signaling were inhibited by ART both in cells and tissues. So these 2 graphs, depict the Western blots, which show the protein level reduction of C4s upon ART treatment. And here at tissue from fibrotic mouse model, where again upon ART treatment, there's reduction at ERK and FOS signal. So to conclude, some key messages, let's say, through high throughput screening using novel screening tools, this group of scientists identified ART as a promising anti-fibrotic compound. Different studies, including in vitro, in vivo, in silico, and single cell analysis, revealed core aspects of ART's anti-fibrotic activity and mechanism of action. TLR4 MD2 complex formation and its downstream signaling arose as a crucial cascade of events during cardiac fibrosis, and the main cell of interest, as this group's name suggests, is fibroblast, highlighting its importance and upgrading its central role during disease. And if we want to see what's molecular changes and how molecularly this compound inhibits this whole process and ameliorate cardiac fibrosis, if we take as a start point the TGF-beta receptor here, upon TGF-beta increase, there's downstream signaling, which through ERK or SMAD, it upregulates some transcription factors, which lead to upregulation of some key fibrotic genes, transforming the fibroblasts into myofibroblasts. This includes many fibrotic characteristics upregulation, including the ECM deposition, which means upregulation of fibronectin on terracing C, two molecules which can interact with this receptor complex, TLR4 and MT2, through a positive feedback loop. And that's where our molecule of interest, our compound of interest, artesunate, comes into play and inhibits the formation of this receptor complex, and so this positive feedback loop, blocking the progress of fibrosis and improving heart function in the end. That's all from us, and we're happy to discuss any aspects or any questions or etc. etc.
Thank you, guys. This was a great sum up of the paper. I know it was very long in its finding and it spanned a lot of different techniques, so that was a very concise way of presenting it. I guess if anyone would like to ask something or comment on something on the paper,
I had a question, if that's all right.
Yeah.
Thanks, guys. Really, really nice breakdown of the paper because they had a lot of stuff in it. Do you mind going back to the single cell RNA-seq graph where you had the kind of pseudotime aspect? So yeah, because I guess my question is, it looks like in most groups you pretty much have all of the cell types present, right? So even in the sham control, you still have these really high, what you would call kind of fibrotic
it's there.
fibroblasts. So I was just wondering if you could comment on that a little bit, and also maybe about the sensitivity of each of these populations to art. Are they all reacting in the same way, or do you think like ahead of time, because they're all present, they might have differential responses to the art based on the expression, I guess, of you're saying the TLR4, for example.
So
the first part, the main population as well. Yeah.
So the first part of the question. So yeah, I think better here we can see where are the populations, so that we have cells there. It's not that in the sun we don't have it. What they discuss in the paper is that these are probably myofibroblasts. So we have cells that express excessive extracellular matrix also in control mice. But I think that this is also has to do that because they talk about population number 1. So this has to do with the resolution that you use in your single nuclei or single cell experiment. So okay, I can see that there is the increase in the proportion, but I think the main point is that these cells they have here that if this, I don't have the graphs because I don't have the data, but if let's say this is the population number one, which is not, but we have the increase. They say that we have the increase in the collagens and the fibrosis related genes, and then we don't have them in the sum. So I don't think that the main point is the increase of the proportion rather than the change in the expression of genes. What was the next? Sorry, second question.
So the next question, I guess, so if that's the case, is the changes in genes in response to art the same across all of those populations, or just in that population one that you were saying?
Nice question. If I had the data, I could say, but I don't know, because what they discuss in the paper is that actually what they say is that this population goes here through pseudotime and then goes back on the treatment. But I don't know what happens in all these different populations, if they are different populations. You know, you can understand what I mean. You know, this is resolution in a single cell experiment, so it's not like it's defined different populations.
Yeah.
So, I don't know if they are different.
Because I'm just trying to get a sense of how art like works, in the sense of when they do their in vivo models, they kind of do the daily injections of art, I think you were saying. So is it just really keeping like a specific pool of fibroblasts at bay, so not allowing them to become this really high myofibroblatic type thing?
Yeah, what I understood is that through inhibiting the formation of the TLR4 and MD2, you cut the feedback loop that Filippos talked about, so you don't let fibroblasts become myofibroblasts, or you don't let them express higher genes related to fibrosis. So I think this is the point, because it's not like they say we have a new subtype, or I think it's just the change that the expression of specific genes like collagens and fibronectin and periostin just changes. So when you add art, you block this pathway, you block this feedback loop of dumps. So you block this transition or this change in the expression.
Cool, thanks.
I mean, if I may, I think it was in this single nucleus RNA-seq that we're discussing, I think I had a question like, I was thinking maybe something along the lines of what Harry was saying, because I was wondering why they used that preventative treatment model for their single cell and not their therapeutic one. It's not the mice that they first performed TAC on and then gave ART to. It's the combined treatment, like a preventative regimen that they did single cell on. So I was wondering,
probably, if I guess, they probably have done it.
Yeah.
But maybe they don't have the same results.
Yeah, I
don't think that they have done all this work and they didn't do a single nuclei in another model. So probably this is the one that works, and the pseudotime has this really nice loop. So
yeah, very good. Is there anything anyone else would like to comment or say anything on the paper?
Yeah, hi. If I may comment on something. I was really kind of the fact that they couldn't find these macrophages, and then I kind of also went into the supplement and was looking at why did they say that they don't have macrophages in the data, and is it truly that it's only based on that there wasn't a change in the macrophages? Was it so?
Yeah, they have macrophages, but the proportion doesn't. I think if I remember well, it's about 20%, 20-25%, and it doesn't change. This is what they say, because I think it was a question, because this model is also, inflammation that turns to fibrosis. So this is, so it was a question about what happens with inflammation. So they say that it doesn't change. But I would really like to see what changes because in the cardiac fibrosis we have a lot of talk through macrophages and fibroblasts. So I would really like to see what changes in the macrophages because I'm sure something's going to change.
Yes, that would have been my kind of follow-up, that if the drug
sorry.
is now targeting TLR4, so I guess one would assume that it has to then, I mean, it's not selectively going only into the cardiac fibroblasts. It's going to target everything all around everywhere. And especially, I think maybe in the discussion, they were discussing that if you give this drug for a long term, then what might be the side effects with macrophages and everything else. So what do you think about that, or do you have any ideas?
Maybe if I can comment something on this. So they state that what is known based on previous research is that ART can act through TLR4 on macrophages upon LPS. So this is a known interaction. Although the fact that the populations of macrophages don't change maybe it doesn't implement that it doesn't have or it couldn't have activity on macrophages, but maybe it's something that they at this moment they do not interested at. Don't know.
Yeah, also that the thing is that if you don't see a change in the proportion in a single cell experiment, it's not like you probably have changes, but it's not that single cell also captures all the changes in the proportions in every experiment and every tissue. So it's just one explanation or just an image of that it doesn't change in the single cell. It's not, maybe they change, but it will be interesting to see what changes in the expression level. I think this is the nice thing here.
I think
Yes,
Ruth
definitely.
has a very nice point here, because they even said when they were trying to identify this target of ART that they even mentioned it as a reference that in macrophages ART inhibits TLR4, or it is known as to have an anti-inflammatory effect. So I think it was a very nice point. What would happen if they used it long term? Because I think the longest that they used it in their mouse models was 4 weeks, I think, but they didn't go in to check on what happens with immunity there. So very good point.
Yeah, but in this model, I think maybe this is one of the reasons that they choose the preventive one, is that this model goes from inflammation to fibrosis. So I'm not sure that 4 weeks after that they start the therapeutic one, they still have inflammation there. Probably it's fibrosis that gets worse and worse and worse after 4 weeks. So this is also a difference in these two models. So in 4 weeks that you give the ART, you also have the inflammation plays a really strong part in this time of the model.
Yeah. Just a comment on the other single cell data that you were just showing. I find it kind of peculiar that they say that they have these 6 fibroblast subpopulations, but yet they don't name them or show any markers how they define these populations. Because with single cell, how you cluster, I mean, you can get a lot of populations or not, but I'm just kind of surprised that in a journal of this level that they don't have very details about how they are identifying clusters or stuff like that. So that was just a surprise.
I'm not that into the fibroblasts in the heart literature, but I believe this is just the resolution that worked best. So this is why it's not that they have 6 different subpopulations. Maybe they do.
Next paper they will come out with all the identities and everything.
Other questions?
May I? First of all, can you hear me?
Yeah.
Okay, perfect. So it's more like points of discussion. So it has to do with what Ruth said before, that this population is actually the same as the, how's it called, trajectory analysis. So usually when you do a subclustering and a trajectory analysis, the human plots are different. So it's like the subclustering and the trajectory analysis are overlapping. So I don't know if these are true subpopulations of fibroblasts, since they don't name them and they don't have any marker expression. Something peculiar happens here. As a point of discussion, more like we discussed it also in the previous paper, the thing with the trajectory analysis. And the second thing was in their in vivo models, I don't know how it affects the healthy tissue as well. So you have the fibrotic part and how it affects fibrosis, but you don't know how it affects healthy fibroblasts in vivo. And also the effectiveness. I mean, you give it, you stop the positive feedback loop, but you haven't stopped the TGF signaling. So if you stop giving the drug, what happens next? Will it not create fibrosis again? Just as a point of discussion, not like.
Kostas, I don't know if you're talking. I think we cannot hear you.
Now? Yeah, now you can hear. Okay. So it's not only TGF, but in a tissue, right? So I don't know. I think this is just a suggestion with a feedback loop, and only because they need the dams. Some of the extracellular matrix proteins, they act as dams to activate TLR4. But yeah, I don't know what happens to the healthy tissue, and if this also, I don't know, in the healthy fibroblast we have the complex.
Yes, but I suppose so. But what they say is that what they found is that at the diseased cardiac fibroblast, they have found that the ECM components interacting with this complex were much upregulated compared to the healthy ones isolated from the isolated biopsies. So I suppose that you have some basal expression of this complex at the healthy tissue. So maybe this compound can act maybe at a very minimum level or slash non-active level. I don't know. While when you have this overexpression of formation of this complex, the compound has the ability to interact more and exert its effects more robust compared to the healthy tissue. I mean, that's in the more general rule, I think, of how a compound would act. I don't expect zero effects in even healthy tissues, but the whole idea is to have some therapeutic effect. That's my point of view.
I think this is a really nice point raised by Christina as well. I think maybe the point along the fibrosis, timeline would be better. It would be different for this one, since if we take their reasoning for it, it's ECM related dumps that activate the TLR4. So I guess that would require some level of fibrosis first in order to develop these dumps, and then the therapeutic potential of ART could become beneficial.
But you have dams because you have the inflammation there. So you have dead cells, so you have tons of dams in the tissue. So it's not like that you need just fibrosis to turn on the loop. You just need dams. So with inflammation, you have destruction of the tissue. So you have tons of dams to turn on this loop.
That's true. I'm just, I guess, equally surprised as Christina and Harry that they even, make a point of it not downregulating SMAD2 signaling on the TGF downstream of the TGF receptor. So I guess I would have liked to have seen like how those two would interact. But it's a very nice, out of the box, I would say, approach into drug repurposing, because ART, as they discuss as well, is an anti-malarial drug which acts completely differently. So it was a really nice repurposing, let's say, drug repurposing paper, and their thinking was quite interesting. So
I can say the question if you know. I can say something, I can state something which I didn't get. I discussed it with Costas, and maybe someone can have an explanation of why they do study this concept here this way. So what they say, so they say that we're interested in TGF beta stimulation in cardiac fibroblasts, etc. So we don't really care about the LPS effect on fibroblasts because maybe it involves more inflammation, not fibrosis. So in vitro studies and their single cells, etc., they are with TGF beta stimulation. But when they do study the binding potential of this compound in silico, they do it with LPS. So I didn't get how you might be interested in studying what happens upon TGF signaling, and you want to study and support your concept by showing something with LPS binding, like so they study the LPS and then they go with TGF everywhere, apart from HEC, which is so here the LPS treated cell are HEC, while the TGF beta treated cell are the cardiac fibroblasts at which we're interested in. I don't know if I made my point here, but it's something that I couldn't understand the experimental their experimental thinking on this thing. Maybe I confused you.
I mean, I don't know if any other dumb structure for this kind of stimulation is available, because LPS is quite common, so I'm sure they can use it in their stimulation, but that's actually quite interesting why they use LPS for HEC. I guess.
They use so what they say is that in fibroblasts during fibrosis, this complex, okay, we believe that it interacts with some ECM components such as fibronectin 1 or tenacin C, which are upregulated upon TGF, and that's how this feedback loop is generated upon TGF beta signaling. And they go and they study this interaction of ART upon LPS binding in silico. So I don't get this. Why don't they study upon fibronectin 1 binding, for example?
Yeah, I guess I don't know if
Anyway,
I have no clue, because I don't run molecular interactions or all atom interactions, but I don't know if LPS, its structure for these kinds of experiments is very well known and well studied. So I don't know if the ECM dump structure is available or anything to run these kinds of interactions. But
I suppose fibronectin structure should be available. I don't know. Anyway, it was one of my worries in this bit here. I couldn't answer it myself.
Maybe I could just make a little comment. I think when they do these structural assays, they were just curious to see where the binding site of the drug, the ART drug, would be in comparison to a known drug, the actual MD2 inhibitor, L6H21, as well as to the natural ligand, which is LPS. I know in pharmacological studies, they always like to use a ligand with the drug, and they say that if the drug is able to have an effect without the ligand, that it's not a canonical effect or like an effect directly blocking it. So I think that's what they might be trying to show in this in I here. So I think that's why they did this particular experiment as well with SPR, which just tells you receptor ligand binding or drug receptor affinity as well. And I think the reason they kept TGF-beta for all of them is because it's just the way to model the fibrosis. And I think they were just trying to show that this drug can inhibit fibrosis with or without the natural ligand, which would have been LPS. And also it works equivalently as well as the MD2 inhibitor in terms of inhibiting the effect of LPS.
Yeah, this could be an explanation. Thank you.
Yes, I think Tamin so like really put this into perspective, because I think we're really going after these CM dumps, but maybe it's, you know, just to prove like the concept of this study. So that is a nice touch by Jamin. And I think the meeting will end soon anyway, but is there anything else, any other comment one of you might have on the paper? Any final words? Okay, so once again, thank you very much, Kostas and Filippos. This was a really, really good presentation, and thank you everyone for the discussion. We have our next meeting planned for March 10th. Follow us on social media, so we will be announcing more information on that meeting soon.